Good morning, good afternoon, and good evening to our global audience. Thank you for joining us today and welcome to our webinar, The Advantages of Kinome Profiling with a Continuous Assay. My name is Nicole Heduk and I'm Digital Project Manager at Assay. Before we begin, I would like to take a moment on some housekeeping items. Ahead of today's presentation, we will take a second to introduce you to our moderator and presenter. After the presentation, we will conclude with a Q&A session. And for further information, please visit our website, assayquant.com, and follow us on LinkedIn for all the latest information about our technology. If you have additional questions or would like to schedule a meeting with members of our team to discuss your science personally, please email us at hello@assayquant.com. Now, I will introduce you to your moderator today, Kelsey Schwartz. Kelsey is our marketing manager here at Assay. Kelsey received her Ph.D. from Northwestern University and has over a decade of experience in the digital marketing space. Kelsey, I turn it over to you.
Hi, everyone. Welcome. Like Nicole said, I'll be helping moderate the Q&A today and help get all of your questions answered. Feel free to submit your questions at any time during the presentation. We can see if we can sneak in a few while Earl is presenting. Otherwise, we'll save them all for the end. While you have the option to send your questions anonymously, we encourage you to include your name because if at any point we run out of time or are unable to answer your question, we can definitely follow up with you and make sure your question gets answered later. We're very excited to share this webinar today. We'll be hearing from Dr. Earl May, Assay Senior Director of Discovery Technologies. Earl brings us over 25 years of expertise in drug discovery and enzymology from several biotech companies. He's explored hundreds of drug targets, including many kinase inhibitors with both reversible and covalent compounds. He's been a longtime user of our SOC sensor peptide technology, and we're very excited for him to share his expertise with all of us today. Earl, you can take it away.
Sure. Thanks, Kelsey. Thanks, Nicole. Let me share my screen here and put this, if you don't mind, over here. There. Can you see my screen? All right. Well, thanks everybody for joining us today. We're going to be talking today about just what our Kinome profiling assay can do because it's a continuous assay and what it can offer over the traditional endpoint assays, which are the mainstay of Kinome profiling out there. So today I'll be talking about, I'll open up with an introduction into the difference between continuous assays and endpoint assays. I'll then introduce you to our PhosphoSense platform, and then introduce you in particular to how we do Kinome profiling and what our data look like. Then I'm going to get a little bit into the nitty-gritty. I'm going to tell you about a very important part of what we do, and that's initial rate determinations, which can get very important when you're screening many compounds over many kinases. And then we're going to get into what these advantages actually are. I'm going to do that by introducing you to Francis Anscombe, a dynamite statistician who basically made his name on, well, on a lot of things, but what we know him as is the champion of always look at your raw data. And then we're going to translate that into always look at your raw data for Kinome profiling by going from Anscombe's quartet to Assay quartet. At that point, I'll wrap it up in a summary, and if everybody's good, I might throw a little teaser at the end into what might be our next webinar about time-dependent inhibition. So without any further ado, let's talk about continuous and endpoint assays. Everybody's worked with endpoint assays. You start with an assay that you've very well characterized. You've shown that it's linear with time. Hopefully, you've shown that it's linear with enzyme concentration. You've done all your characterization. You know what's going on. And at the end of that endpoint assay, you get a point. Or if you're doing a duplicate, you're getting 2 points. All right. But that point is simply the signal at that time point. And the assumption is that it's linear from the zero out to that time point. And so therefore that point is actually a reflective of the initial rate of the reaction. So that's actually dynamite, because if you have an awful lot of endpoint reactions, you can stack up all your plates if you're doing thousands and thousands of reactions like you would in a high throughput screen, and then your reader can just fly through them like that. However, there are disadvantages to this. There are a lot of your critical assumptions, that assumption that that single time point reflects the initial rate, is very often wrong, especially when you're trying to assay, hundreds and hundreds of kinases at the same time. It's very unlikely the hundreds of kinases that you're looking at in a kinome profile are all behaving very, very well. All right, so you are also only getting one or maybe two reads. Okay, and so you've got a lower confidence in what you're actually measuring there. Maybe you'll get the average of two reads, and that's as much confidence as you have in that. So that right there is a reflection of the of your precision in the assay, whereas your critical assumption being wrong is a reflection of the lack of accuracy in your assay. So, and finally, that if it does deviate from a straight line, it's very possible that there's some really important critical mode of action information that's buried in there that if you're doing an endpoint assay, you're never going to see. All right. Contrast that with a continuous assay format. Now we're looking over the entire full reaction progress curve from the beginning to the end. Right? And if everything's behaving perfectly, as it would be for your assumptions for an endpoint assay, you get a straight line right from the origin out as far as you go here. So if you have a 60-minute time point, that would be your read for your endpoint assay here. All right, so there's lots of advantages to this. Using a continuous assay format greatly makes it a lot easier to develop your assays, optimize them, and then finally characterize them. You actually have because it's a continuous assay, you've got this internal control for any kind of behavior issues that might crop up during the assay, any curviness in a line, any temperature differences, anything like that, readily apparent in your assay here. Because you've got lots and lots of data points, all of them contributing to that initial rate, you've got a lot more confidence in that rate that you're talking about there. So therefore, you're getting much more precise data. And you can detect interesting and exploitable modes of action, which may mean that the final rate that you're getting from the endpoint assay may not be the best rate assumption for what you see in the continuous assay format. And that's a reflection of the accuracy of your assay. So there are some disadvantages. You need to actually, if you're doing a continuous assay, you have to read it over time. So you got a lot more need for readers in all of this. And also, it's not just one read. You're reading all the way through the reaction, and you have to do some really sophisticated data analysis to extract some initial rates from the thousands and thousands of reactions that you're doing all at the same time on your microtiter plates. So with that, I'm going to talk about the continuous assay that we offer here, and that is, before I get into that, let me just summarize with our so with your endpoint assay, you've got low accuracy and low precision both. So you're not getting clustering, and that clustering might not be on the bullseye. But with a continuous assay, you have a much stronger chance of both getting high accuracy and high precision hitting the bullseye. So at PhosphoSense, at AssayQuant, we offer the PhosphoSense continuous assay format. And this was developed initially out of the lab of Barbara Imperiali, where a chromophore here, the sulfonamidoxine here, is actually derivatized to a peptide substrate proximal to the hydroxyl that will ultimately be phosphorylated by your protein kinase. And when this hydroxyl is phosphorylated, then that will chelate a magnesium between the phosphate and the SOX residue, the sulfonamidoxine SOX residue. And that chelation results in an increase in fluorescence here. So we excite at 360, the emissions we read at 485, and this can be done continuously, just like that, so that in the end, you end up with a progress curve in every well. And here we're showing a 384 well plate. We started in 96. We're working in 1536 at this point. So a progress curve in every well. You can do dose responses. You can do a whole variety of applications, which you'll see in a second. But all of this is dependent upon this chelation enhanced fluorescence that's made possible due to this SOx chromophore. So it's direct, it's homogeneous, and it's most importantly continuous. It can use a wide variety of ATP concentrations, all the way from below km up to millimolar concentrations, which are physiological. This is a very simple assay to read. You have the right filters and mirrors in your reader, which are very commonly available. You can do it in your lab, and it works with any plate type. All of this is covered by patents from MIT and exclusively licensed by Assay Quant. It's simple, it's powerful, and it's very flexible. So in addition to you can do your reaction with magnesium, but then if you add europium, you now have an endpoint assay. Okay, and what's more, you're doing the reaction with magnesium to get this continuous read, if you're interested, and adding in europium actually redshifts the assay and introduces time resolved fluorescence into this. The advantage being any compounds that may fluorescently interfere, this does a great job of eliminating those from your reads here. So I show this just to say that we actually have two formats here, a continuous assay format, and then if you spike in europium, an endpoint assay. So you can actually use this chelation enhanced fluorescence as a method of both doing the kinetic read and situations where you want to do many plates at the same time and don't have the readers, like high throughput screening or many tests for SAR. You can use the europium format, the red format. So this is actually a game changer here. What we do at Assay Quant, we have a whole variety of kits and bulk reagents that we can supply for over 500 assays that we have available now. And so those will enable you to do those in your lab. If our kinase, your kinase of interest is not available, we can actually develop those assays for that kinase in-house, and we have a very good track record of delivering on those, partly based on the fact that we've been doing this for a while. So we have over 35,000 sensor peptides in our bank right now, and we do an awful lot of work making sure that these sequences are related to the substrates of your kinases of interest. So we will work with you to develop these assays. And if you're not in the position where you can do these assays in-house, or you're interested in some more sophisticated type of analyses, we do custom compound testing services in-house. That includes a variety of potency testing, from simple IC50s on through time-dependent inhibition, traditional mode of inhibition analysis, activation studies, and most importantly, profiling studies, which is difficult for folks to do unless they have all of this set up in their lab. So here are the various applications that we can do here. We do assay development in several rounds. We call them milestones. We have a very good track record of developing good assays for your kinases. When we're done with that, we actually can fully characterize that assay, developing all of the Michaelis-Menten type parameters. We can do mode of inhibition analysis with your inhibitors to determine competitive, non-competitive, uncompetitive. If it is an activating inhibitor, we can set up the reaction in such a way that we monitor that. Where you add the inhibitor, you see an increase in the rate like this. We did have a whole variety of ways of gauging the potencies of your compounds, from simple IC50s and KIs through resonance time and K-inactin Ki analysis. And then very exciting, we are developing sensor peptides that can be exquisitely specific to your kinase of interest. And what that allows us to do is actually do our assay with cell or tissue lysates, and then be able to have some confidence that the signal that we get in that assay is due to the effect of your inhibitor on your kinase of interest in the cell lysate itself. And this opens the door to biomarkers and companion diagnostics. It's something we're very excited about. But really, the topic of conversation today is kinome profiling, and that's what I'm going to spend the rest of the talk talking about here. So we have 403 wild-type kinases at current, actually a little bit less. And that's a good fraction of the 518 some kinases that have been identified out there. And we're adding more and more as we go along. As I said, what we do here is direct and continuous with high accuracy and high precision, all from the assay system itself. And what we do allows us not only to get an endpoint, but really to, if we're in the sweet spot of the inhibition, we can actually diagnose time-dependent inhibition and enhanced lag, other types of interesting kinetic observations right from the initial reads at the start. We're very flexible with ATP, unlike other kinome profiling platforms. We can do it at km, we can do it at 1 millimolar or physiological km, or we can do them both in parallel to provide initial mode of inhibition type information. In addition to ATP, there's lots of everything else we do here, we can do at physiological conditions, starting with the substrate sequence itself, which we strive to be as close to physiologically relevant as possible. And we use physiological levels of our ions and everything's optimized for each individual target kinase independently. So our kinase panel is each kinase is optimized independently, and is we check multiple sources of kinases from different vendors to really make sure we're looking at the best one for our purposes. There's just this profile, this platform, this technology that we do for the entire panel. Some other folks out there, they'll do different types of assays. They'll even mix enzymatic assays with biophysical assays. This is all phosphosense that we're doing here. And as I said, we're adding more and more assays as we go along. So here's some examples of what we can see for this. Every month, we'd be sure to run at least one staurosporin control, and that helps us make sure that all of our kinases are behaving properly here. If you do staurosporin at 0.1 micromolar with ATP at km, you can see that you see 179 of our 400 some kinases will light up with percent inhibition values greater than 70%. As would be expected for an ATP competitive inhibitor like staurosporin, if you raise the ATP concentration to physiological to 1 millimolar, then you're going to see many of these percent inhibition values drop as the ATP washes out the inhibitors in here and in a competitive displacement assay, a competitive displacement method. And you can see that those kinases with higher KMs are more readily washed out. And so at 1 millimolar ATP, you have only 78 hits that are greater than 70% inhibition. When you actually look at some test compounds here, you can see it range from, here's ibrutinib, which largely lights up the tyrosine kinase family. You can see a variety of these things lighting up here, but largely in the tyrosine kinase family, or here's one hit in tyrosine kinase-like family, or perhaps BI4020, which does light up the HER family, but also has representatives in various other families, including the CMGC family, the MAPKK family, CK1 family, and even one down in AGC land down here. So it doesn't have to be all within the same family. As things get more and more selective, here's a compound which only inhibits a few of the TEC inhibitors in the TK family. And then finally on the right here, here's a client who gave us a compound which only hits HER2 when it's present at 1 micromolar in the assays. And with 1 millimolar ATP, you'll only see one that's greater than 70% inhibition. So you can see some remarkable selectivity come out of this assay. So lots of assays, lots of compounds, lots of kinases. What makes this different from more difficult for endpoint assays is that we have to get all of those initial rates. All right. And you might remember from your original chemistry or biochemistry classes, you might follow an enzyme's activity and you might pull out your ruler and draw a line based on this and then determine the slope, and you'd call that the initial rate of the reaction. That is very subject to error, not only user to user, but enzyme to enzyme type differences. And frankly, it just takes too long. So what we need is a fully automated system that allows us to have consistency between the enzymes from experiment to experiment, from user to user, and allow us to crunch the data that comes out of all of these plates at the same time. So what we have developed an automated system now that allows us to find the linear range. It allows us to avoid the lag part, avoid any potential saturation that might be happening, and really zoom in on this linear portion here. And what's really important is that, you know, for kinome profiling alone that we do here now, we have 34 plates, 384 plates per day that we can do. And that's over 10,000 progress curves a day. And we're actually in the process of incorporating 1536 well capability. So pulling out your straight edge is not going to be really helpful here. We need something automated. And so we actually came up, and by we, a very talented associate, Sam Huare, with a summer intern, Tony Keenan's help, we came up with an algorithm that allows us to, in an automatic fashion, find and report the linear range. It is actually a lot easier than it looks here. I'm just going to walk through it very quickly. Basically, the first thing you do is you track up the line, and you'd say, I'm looking for 10 consecutive data points up this line, and I want to find the maximum slope. All right, that doesn't take long. Just track up, find the maximum slope for those 10 points, and you say, okay, there it is. That's the maximum slope. You find out the error in that slope that you have, and then you find the three times that error, the 3 sigma, you determine that as well. And then what you do is you start at that linear portion that had the maximum slope, and you start creeping up the line from there until the error in your slope goes beyond that 3 sigma point that you found. So when it's starting to deviate significantly from linear, then you found the top point of your linear range, and you mark that. And then you go in the other direction, and you do the same thing, and you find which point as you go down is the one that deviates from linearity beyond that what you've determined, and that's your bottom point. And you go from bottom to top point, that's your linear range, that's your slope. And we can do this remarkably quickly and process all of our data very quickly, and allow us to then pop that into those initial rates into our spreadsheets and into our data management system, such that we can then use that to do everything else that we do. So here are some examples of what we find. If everything's behaving nicely, like it is for Axel here, you can see a very nice linear range here. When you go bottom, it goes almost all the way to the bottom. Here, it's starting to deviate and plateau a little bit, so that's the linear range it came up with. With this BTK progress curve, it's plateauing off more quickly, so you only get this portion for the linear range. Here for CSK, you see a very significant lag. And this actually goes up and finds that this is the maximum rate. It goes up and determines, okay, that's the top, but when you get down to here, that's the bottom. So even though you're an hour displaced into the reaction, this is the rate that it will pull. All right. Same way for FGF4, a slightly smaller bit of lag here. For a noisier reaction, like we see for HER2 here, it does a nice job of still avoiding the lag, avoiding saturation, and giving us a fairly good linear range, which might be impossible in an endpoint assay because it might be so noisy. More about that in a bit. And then here's HER2 with, again, a very slight lag that you see here, but then this is determined to be the best point that we see, the best range that we see here. So this is what works well for us. Now we're interested in looking at the data that we get from our kinome profiling. And here's where the fun starts. I'm going to introduce you guys to a wonderful guy named Francis Anscombe. If you guys haven't heard of him before, let me tell you a little bit about him. He actually he's Cambridge educated. He was a lecturer in Cambridge from 1948 to 1956, and then he went across the pond, joined the faculty of Princeton, and then stayed there until 1963 when he went to Yale and set up the statistics department there, and then did all these wonderful things. He has a very rich set of contributions that he's made to the field of statistics all the way up until he passed away in 2001. Now, the biggest thing he did was he was right there when computers came in. And so he was integrating statistics into computers. And in 1973, one of the things he was trying to stress to everybody is that computers are great, they're cool, but you should use them to both do your calculations and make graphs. All right. In other words, always look at the raw data. And to drive that point home, he came up with this really wonderful demonstration that he published in 1973 of Anscombe's quartet. And so what this is, it's it's all right here. It's 4 data sets, each with 10 pairs of XY data. All right. These all have identical average X's and average Y's. They all have the same linear regression formula and the same confidence measures. But when you look at the data itself, it's clear that if you only look at those numbers, you're going to miss something significant. So here's Anscombe's quartet. You can see that there are 10 points in each of these sets, 10 XY values. The average of all the X's is 9 for each of these 4 data sets. The average of the Y's is 7.5, and the standard deviations are the same across all 4 sets for both X and Y. Okay, if you do a linear regression for each of these data sets, you get exactly the same slope, exactly the same Y-intercept, and exactly the same mediocre correlation coefficient of 0.67 here. All right. So let's look at these data. All right. Here's the first data set. This is kind of what you would expect to see. You'd expect to see a rough line through this. It's not great. Your correlation coefficient is fairly low. There is a significant linearity in this data set right here. This is kind of what you would expect. Now we're going to look at data set B. Okay. Now you see something very systematic happening. Okay, it's not noisy like this, but clearly there's not something linear in these data points. Something's going on, and you may not have any idea about it. And if you were just looking at these values up here, then you would have no idea that you weren't looking at something like this. You were actually looking at this data set C. You've got a nice straight line like this, but you've got a point that's all the way up here. So your Y-intercept is actually negative. Your slope is significantly lower, but you might think than this data set here, but it's all offset because of this one outlier. All right, but still has the same numbers. And by the same token, here's data set D. Everything's 8 except for this. This, if you crunch the statistics, it looks exactly the same as the other 4 data sets. Now, if you were to plot the residuals for all of these. This would all look random from here, and there'd be something very systematic about the residuals for these here, especially this one. You'd see as something increasing, you'd see a blip here and here for these, or as you go out along that line that you would get there. So the advent of residual plotting that we all do nowadays would help with this, but you still have to look at that data. If you look at this data, you're going to say, this is not what you expected. All right. So that's basically what Francis said here. He basically said, always look at the raw data. And this is very important. We all know the medicinal chemists out there who are interested in that number in the box, but we as the folks supplying this biochemical information need to provide the context to help them guide the SAR. And that means always looking at the raw data. So around this time, we were developing a poster on what the advantages of our continuous assay format are here. And a very talented associate in our group here, Matt Hakar, I tasked him for looking for examples of good progress curves that we could put on this poster. And he said, well, wait a second here. I liked what I heard about Enscombe here. We could actually tell the same tale with our data here. And so I said, okay, that's cool, Matt. Go out and get the data. He got the data, and here it is. So I'm proud to present Assay Quant's quartet here of data. So what we have here are the endpoints, okay, for the Kinome profiling progress curves that we have. We always do our things in replicate in our progress curve. So here's the first replicate. Here's the second replicate without inhibitor. Here's the first and second replicate with the inhibitor at its concentration for 4 different kinases here. Okay, so this is actually the endpoint that we have here. And if you actually do the math, they all kind of turn out the same. They're all roughly 65% inhibition for these 4 particular kinases in our profile. Okay, so they're all roughly the same, right? But let's look at the progress curves and see how these curves lead up to these endpoints and see that there really is some additional information that becomes readily available because we're doing a continuous assay. So let's do what Frank says. Let's look at the raw data, all right? For TTBK1, it is exactly what we would expect. We have a really nice progress curve. Here's the uninhibited with the inhibited reaction. We see a drop down, so it's 65% inhibition. This rate is 65% of the uninhibited rate. Now, this is great. This is what we hope to see for if we were trusting an endpoint assay, but we have so much more data that gives us so much more confidence in these numbers here. So, that's the first example. For FGFR3, here's an example where things can be noisy and it's not readily apparent in your endpoint assay. Now, if you look at the average deviation when you add the inhibitor, it's actually the error is significantly higher here. But if you hadn't noticed that, you might be inclined to think the 61% is assured in all of this. But with the continuous assay, you can see it's noisy. It's nice and continuous at the very beginning. It gets noisier and noisier and noisier. That's got all the hallmarks of something that's crashing out of solution. All right. Then the fun starts. Okay. With BTK, all right, here's an inhibitor of BTK that actually shows a whole bunch of saturation as you get deeper and deeper into the progress curve here. So the level of inhibition is dependent upon the time at which you read it. That's time-dependent inhibition, okay? And if you can identify that and fully characterize it, you can provide an awful lot of good information about both irreversible inhibitors, of which this is one, or slow off-rate inhibitors. And that early detection of that mode of action, you're just not going to get it from an endpoint assay where you're only looking at this and assume it's 61% that your inhibition. Here, your initial rate is practically the same as uninhibited. Your final rate is 0. So this requires a more sophisticated analysis to actually give you an indication of what's going on. And then finally, here's something that we've actually identified relatively recently. We can see that there's a lag in the inhibited reaction, where we didn't see a lag or saw a much smaller lag in the uninhibited reaction. And as you add more and more inhibitor, you might see this lag increase more and more. All right, that compound-dependent lag extension is a very interesting mode of action. We confess we don't know 100% why and what the source of this is, but we're fairly sure it's interesting. We have some hypotheses. It could be related to the ability of the compound to bind to the active versus the inactive forms of the enzyme. It could also be related to the ability of the kinase to bind to different oligomeric states of the enzyme. So just a few more examples of what these things can look like. We have, here's actually enzymatic activation, where you can see that adding your compound, what you thought was an inhibitor, actually stimulates the reaction rate. And here we would recommend doing some follow-up EC50 analysis on this. Here's another example of noise, and it can be solubility, but there can also be fluorescence interference type issues going on as well. Here's an example of something that shows an initial lag, and then there's a actual increase in rate. We can see these reproducibly sometimes. We make theories, and we don't pretend to fully understand that. If you get this on your readers, we encourage you to definitely do some follow-on studies, because something odd is going on. But really where it's at is time-dependent inhibition and the lag extension that we've been talking about. Not all of our time-dependent inhibition is complete. I showed you an irreversible BTK inhibitor on the previous slide. Here's something that looks like it has a final steady-state non-zero velocity. That is very possible if you're looking at a slow off-rate inhibitor. In this case, we definitely want to do some further time-dependent inhibition experiments, and if time permits, I'll show you a little bit about that at the end. And then finally, here's a couple examples of compound-specific lag extension. Here's a little bit. And here's something where the rate is practically 0, but then starts to pick up an hour and a half into the reaction. We do see this sometimes reproducibly. Important to note that because these are single concentrations that we're doing in our kinome profiling, we can only detect these interesting kinetic observations here if we're in the sweet, if it's not fully inhibited or if it's not fully uninhibited. So it's not going to be a cure-all. It's not going to be diagnostic for everything we put in there. If your thing is flattening this kinase, we won't know much about the kinetics. And by the same token, if we're well below the IC50 for your inhibitor, we won't be detecting these kinds of things either. But yeah, we routinely make observations, annotations of these progress curve anomalies, the kinetics here, in all of our reports. So in summary, what I've shown you here is that our rate determinations for our continuous assays, we've got lots and lots of points as compared to an endpoint assay, which gives us a lot more confidence in the values that we're reporting. When you actually look at the progress curves, you can see an awful lot of interesting things that can happen in the curvature of the reaction. If it's actually saturating, that could be that could be enzyme instability. It could be depletion of the substrate. It could be product inhibition, but it could be that really interesting time-dependent inhibition, which you can use to diagnose and fully characterize irreversible and slow off-rate inhibitors. And then the lag that you see at the beginning of a reaction could just simply be a result of autoactivation or other oligomerization type steps. We don't know, but if the compound affects that lag, that's something that we can characterize as well. And we would be thrilled to work with you to understand this this observation, because it could very well be more than one thing that we're looking at here. So we've got a lot of good methods now that allow us to do this and accelerate our data analysis and get quickly to the point where endpoint assays start. And we'll have all of those data points from thousands and over 10,000 wells per day and growing dramatically. So right now, we are routinely providing our clients with profiling with a 10-day turnaround time. If we push it, we can do it in 5 days. But right now we're implementing full automation capability and a 1536 well plate ability, and that should allow us to reduce our turnaround time down to 3 days. So you've all been good. So I'm going to give you one last teaser before we close, and that's basically to talk about a bit more about this time-dependent inhibition here. If we see a hint of this in the kinome profiling, or if you give us compounds to actually analyze, then we can take this through our standard workflow for looking at compound potency. And we really use this continuous assay format to give you exactly the kinds of parameters that are most relevant for the evaluation of the potency of your compounds. So how do we do this? We start off by doing simple dose response experiments. And we ask, are those curves linear or are they curvy like this? We do plus and minus per incubation studies and see if there's actually a shift in the IC50s if you pre-incubate the compound with the enzyme versus don't do pre-incubation. All right. And if all of the curves are linear and there's no change in the IC50 with and without inhibition, well, then there's no time-dependent inhibition. We can actually report IC50s, and if we have enough other information, we can report KIs. If, however, it is curvy and we do see some shifts in IC50s, then we're interested in finding out if these are reversible inhibitors or irreversible inhibitors. And the simplest way to do that is with a jump dilution experiment. And that tells us right away if it's a reversible inhibitor or an irreversible inhibitor. If it's a reversible inhibitor, we can do forward progress curve analysis and report the off rate, the half-life, or as is most used nowadays, the residence time of this. And those are all things that you'll want to use to report the potency of your reversible inhibitor, your perhaps slow off-rate inhibitor. For an irreversible inhibitor, we'll do forward progress curve analyses, but this time fit it to something else entirely, and we'll report KInact on KI, or KInact separate from KI. And those are absolutely vital for measuring the potency of irreversible inhibitors. If you are driving your SAR with IC50s for irreversible inhibitors, you should take a close look at what you're doing. If you read a paper that reports IC50s for irreversible inhibitors, you should probably put that paper aside. Okay, so anyway, that's just a sneak peek of what we can do. Really, it's important to note that it's important to note that this is the value of the kinetic data that we have for time-dependent inhibition. At best, you could be missing something important like the residence time here, and by using a continuous assay, you're going to give much more information to drive your SAR. But at worst, you could be greatly misleading if you're only using IC50s rather than using the kinetics of a continuous assay format to characterize irreversible inhibitors. So with that, I'm going to close. We're really looking forward to working with you, and I'll be happy to take any questions if any have come through. Thanks a lot.
All right, thank you so much, Earl. We hit our scheduled time, but we're happy to stay on a few extra minutes and answer the questions we've received. And if you have any more questions, please feel free to put them into the Q&A. So we can start to go through some of the questions we've gotten. We talked a lot about our continuous assays and kinome profiling, but can you talk a little bit about whether you can use the europium endpoint assays for phosphatase reactions?
Oh, absolutely. And that's something actually I left off of this slide here when I was going through it. I had a little note right here, somewhere into here, that really we can look at activation, and there are other, not just kinases. We look at protein phosphatases. We just look at it in the other direction, and we actually have some lipid kinase assays as well here for DGKs, diacylglycerol kinases. But to address your question, can we do the red format for phosphatase reactions as well? Absolutely. We do the reaction. We can follow it kinetically with magnesium. If then we add the europium, we can get that immediate read there. And here it's a signal decrease assay. So you'll see a decrease in the signal, your rate that you'll get with as you add more and more of your inhibitor in all of this. I'm sorry, as the phosphatase activity happens. And if you're actually looking at the endpoint, it's the same thing. You'll follow that signal decrease as well. So yes.
Fantastic. Can you talk a little bit about the advantage of performing kinome profiling at 1 millimolar ATP versus ATP at km?
Certainly, yeah. Kind of address that a little bit here. But yeah, at km, there's two schools of thought here. Many folks are very interested in doing it at physiological concentrations of ATP to try and best mimic what's going on in the cell. And we have physiologically relevant substrates. We have physiologically relevant other reaction components that are in there. If we have physiologically relevant ATP concentration, then we're more likely to see off-target toxicity, off-target reactivity to other kinases that's more reflective of what's going to happen in the cell if we do it at that physiological ATP concentration. On the other hand, if you're interested in the actual enzymology, biochemistry of the target interaction there, then you will get the best range of activities, the best focus on that if you do it at km. All right. And so if you're relatively early in the process, most enzymologists will recommend, and many will insist, that you do it at km to cast the widest net possible. If you have a competitive inhibitor and you're doing it at 1 millimolar, you'll never know that you had an active compound that you could then optimize in some way. And so you'll run it at km. So there are advantages to both. We've had this debate. I've had this debate many times externally. We've had this debate internally. And honestly, what we recommend is doing both. If you've got a new pharmacophore series that you're working with, we strongly recommend doing both to give you an initial indication of the spectrum of your inhibitor and also an initial indication of the mode of inhibition that inhibitor might have.
Great. You've touched on the fact that we're looking at 1536 formats. Can you talk about how we're validating that assay format and what projected schedule would be to move forward with that?
Well, that's something we're very excited about, and it's something we're pushing along in parallel right now, and also competing with the installation of our latest full automation capability right now. We've got it validated to a certain degree already, but will we be able to get this going within the next month or two? Probably not. But we're hoping that by year's end, or hopefully even by the fall, we'll be able to start making some improvements to our Kinome profiling system such that we'll be able to do those 1536 well plates there. And we're also debating exactly what the format will be. Is it just going to be 4 of the 3 to 4 well plates that we use right now? And we're happy to provide detailed information on all of this for anybody who's interested. Or are we going to take advantage of this and get still more information on those 1536 well plates? That's all up in the air. No promises, and I'm sure somebody's going to is throwing popcorn at me right now for saying that we might be able to get this done by the fall.
All right, and then it looks like we have one more question that's come in. Do you ever develop larger protein substrates with the SOX technology?
Oh, that's a question we get an awful lot. It's something we have explored, and I know that some other academics out there have actually explored this as well. It's not something we routinely do. We incorporate our SOX moiety as a cysteine residue, as a cysteine-linked residue during the peptide synthesis. Getting the SOX moiety to exactly where it needs to be on the full-length protein is something tricky. It is something we've talked about. It's something that we might be interested in exploring in the future, but it's not something we've developed just yet.
All right, fantastic. Last call for questions. If anyone has any, we can stay and answer them. If not, thank you so much, Earl. Fantastic talk as always. And thank you everyone for joining. And like Nicole said, feel free to reach out if you want to schedule a meeting with us. Check out our website. Thanks, everyone.
Sounds good. Thanks, everyone.